Thanks, Helena. Oops. Okay, can everybody see that and hear me? Okay, so I present the results today of a study done by Helena Westerdahl and me, Emily O'Connor, here at the University of Lund in Sweden. And this is a study on MHC gene expression patterns across songbirds. So as we've been hearing about in this session already, the MHC molecules play a really important role in defense against pathogens. And that's because they bind and present antigens to T cells for recognition as either self or non-self. And in the case of being recognized as non-self, this initiates the adaptive immune response. And the genes that encode these molecules are incredibly diverse. So there are multiple copies of MHC genes in the vertebrate genome, and the alleles that each gene copy are extremely polymorphic. And as Andrew mentioned, the MHC gene region is the most variable region in the vertebrate genome. And this has been linked to the relationship that we see between MHC diversity and the number of different pathogens that can be recognized. And indeed, pathogens and selection from pathogens can drive up MHC diversity. So MHC genes have been really popular markers for disease resistance in studies of ecology and evolution. And one very common approach is to measure MHC diversity within genomic within the genome. First, about the genetic competence of individuals and species from this. And what these studies, amongst others, have shown us is that there's quite extreme copy number variation across species in terms of MHC gene copy number. So we humans, for example, if we take MHC class 1, we have 3 classical MHC class 1 genes in our genome. But then you see extreme variation and many more copies in other species. So a really extreme example is the Atlantic cod. And some estimates put them as having more than 100 MHC class 1 genes in their genome. And the situation when it comes to copy number variation is particularly interesting when we look across birds. So I've borrowed, again, this figure to the right from a nice study by Treminius done in 2018 on number variation in MHCs across birds. And if we look at this figure, the grey bars are indicating copy number and the colours of the branches show us something about the ancestral copy number. And whereas many species of bird have relatively few MHC class 1 genes, so for example, chickens have 2 classical MHC class 1, many species have quite a few more. And this is particularly the case when you move up into the songbirds. So you won't be able to make out the species names, I don't think, on your screens. But if you can see this region where the branches are kind of greeny, bluey yellows, these are all amongst the songbirds. So there's been this big expansion of MHC genes within songbirds. And that's understandably attracted a lot of interest from us in the research community. And often people, including myself, are looking for adaptive explanations for this variation. And indeed, there is some evidence to suggest that being exposed to more pathogens might lead to species evolving to have more MHC gene copies. But when we think about the potential adaptive explanations for the variation that we see, it's really important to think a bit and try and understand more about how many of these copies may actually be expressed. And I'm demonstrating this here just in this toy example, whereby you have 2 species, one has 5 MHC genes in the genome and the other has 10. Well, if only 5 MHC genes are expressed in both the species, then the adaptive interpretation of that variation is really quite different. So this got us to pondering this question of how many of those MHC class 1 genes that we see across songbirds, where you have many species with very many copies, are actually expressed. And we focus just on MHC class 1 for this. And there's good reason to think that not all very high MHC diversity. And these costs are mediated through an increased risk of autoimmunity or a restriction of the T cell repertoire as a result of removing more T cells to avoid autoimmunity. So there's this concept of optimal MHC diversity, whereby you have high enough MHC diversity to recognize lots of pathogens, but not so high as to risk immunopathology. So with this in mind, we came up with these 3 different scenarios that we thought may exist in terms of the relationship between the number of MHC genes in the genome and the number that are expressed across species where you see very different copy numbers. So the first example here in green is all. And in this scenario, all the MHC gene copies in the genome are being expressed. So perhaps the optimal level of MHC diversity exists in the genome. Another alternative is that some species have more than the optimal number, and optimality is resolved through gene expression. So perhaps only a proportion of the MHC genes are being expressed. So this blue line here, proportion, where the proportion is fairly similar across species. An extension of this is the disproportion scenario in orange here. So in this case, you have species with very high MHC diversity. And when that's the case, you may see a disproportionately low proportion of alleles being expressed. And this could happen if there is a nonlinear increase in the costs of MHC diversity, or if there is some kind of cap on the maximum number of MHC genes that are ever expressed. So with these three relationships in mind, we then decided to look across real world data in order to try and see which of these was best supported in real life. So for this, we compiled a data set, a mixture of data which had come out of studies associated with Helena's lab already, and new species that we added, so species that we collected data for ourselves. And this was to achieve a good phylogenetic spread across this. So we had 13 species in total, and from each of those species, we had multiple individuals from which we took blood samples that allowed us to take both DNA and RNA from the same samples. And we used several different primer pairs on every species. And this was to try and ensure that we're getting all the functional MHC class 1 alleles, so that we believe we've got a good representation, or the full representation there. And now I'm going to switch to talking about numbers of alleles instead of numbers of genes. And that's because there are no locus specific primers for these species. So what we get is an estimate of the total number of alleles, but we can't be entirely certain how many genes they correspond to. The two measures are, of course, related. So we can use the number of MHC alleles here as a kind of rough proxy for the number of genes. So we use the different primers we had on both genomic DNA and complementary DNA, so that will reflect the number of expressed alleles. And then we use a deep sequencing approach so that we could sequence all the different alleles and count them basically in the genomic and the cDNA. And then we use Bayesian phylogenetic mixed models in order to account for the shared ancestry of the species when looking at this relationship. And here is our main results. So on the x-axis here, you've got the number of MHC1 alleles in genomic DNA, and on the y-axis, we've got the number of those that are expressed. Each of the data points is representing a species mean, and they're colored by the taxonomic family that they belong to. The green dashed line is showing the one-to-one relationship, that all scenario. So this is where we would expect the data to be if everything was being expressed. So what will jump out at you is that not all the MHC class 1 alleles that these species have at their disposal are being expressed. And we also found some support for a non-linear decrease in the proportion of expressed alleles, such that species with very high MHC diversity express disproportionately few of them. But you'll probably also notice that this is driven largely by this data point on the right, this brown one, and that is the European robin, which has on average 44 MHC class 1 alleles in their genomic DNA and express less than half of them, or fewer than half of them. So if we remove the European robin from the analysis, you see that the statistical support for that quadratic relationship disappears. So what we can say from this is that we can be fairly confident that in the species with low to intermediate MHC diversity, we see a linear relationship between the number of MHC alleles and those that are expressed. But we're less certain when it comes to the species with very many copies with that extreme MHC diversity. It may be that it's just the robins which are strange, or it might be that this is a general feature, this disproportion scenario is a general feature of species with very high MHC diversity. So overall, we've shown that not all the MHC class 1 alleles are expressed in these songbirds, which are these highly duplicated MHC1 genes, and suggestive evidence that species with extremely high MHC diversity may express disproportionately few of their alleles, but we would need more species to confirm that. And we can say that there is a possibility then that optimality is resolved through gene expression, whereby species that possess more MHC genes than the optimal number express just the optimal number. And also an important take-home message from this is that for studies which focus on measuring MHC diversity purely from genomic DNA, they may want to consider that that could potentially be an overestimation of the functional MHC diversity. And then I'll just finish up by pointing you and anyone who's interested in the direction of the citation for the full article on this, for anyone that would like to read it, and to thank the people that contributed data to the study and the funding bodies who made it possible also. And thank you for listening.